Hello, everyone. Welcome back to the third video for your revision classes. So the DNA, which is the genetic material, is going to be where we're starting from today. So which experiments actually led to the discovery of DNA and which led to the structure of the DNA? So all the story about the DNA and the discovery is because the scientists had a thought that was a protein or DNA that was responsible for storing genetic materials in the body. So they knew we have proteins in the body, they knew we had DNA in the cell, but they were not actually sure which of these two was responsible. So this is what brought about these experiments, which has been done, which we're going to mention in the slides to come. So a nucleic acid is a complex biomolecule that stores cellular information in the form of a code, in the form of CTGA, TCGA, in codes like that. So among the scientists which actually did a great work is Frederick Griffith. Frederick Griffith, he carried out experiments that led to the conclusion that a substance can transform bacteria. You need to be very mindful about the discoveries of each of the scientists. So Frederick Griffith, he carried out the experiment that led him to conclude that a substance can transform bacteria. So a substance can transform bacteria from the one which is not causing disease to a disease causing one. So this experiment set a stage for the search for identity of the transforming factor. So we want to know, okay, we know now that that a substance can transform bacteria. But what is that substance that can transform the bacteria? So this is what brought about the research for it. In Griffith's transformation experiment, Griffith actually got two strains of bacteria. So one is smooth strain, which causes disease, and one is a rough strain, which does not cause disease. So Griffith injected the smooth strain, which causes disease, into the mouse. The mouse died. When he injected the R strain, which is not causing disease, the mouse did not die. So Griffith decided to hit the one which is the disease causing the smooth strain. He decided to hit it. So after hitting it, he noticed that the heat actually killed the bacteria. So he made the mouse to become alive even after injection. But when he combined the heated one with a non-disease causing strain, he noticed the mouse dies. So this brought about the conclusion, which has been done by Oswald Avery. So he identified the molecule that transformed the R strain of the S pneumonia, which is the bacteria which was used in the experiment, into the S strain. So what transformed the disease, not disease causing strain to disease causing strain, was a specific molecule. Avery also reached a conclusion when the S cell were killed, it said the DNA was released. So the meaning is when the S strain was being killed, DNA got released from it. So when it got mixed with the R strain, so the DNA got into the R strain and it changed the R strain to become the S strain, which causes disease and made the mouse to die. The R bacteria incorporated the DNA into their own cells and changed into the S cells. Now, there was another discovery which made us to understand or discover the DNA, which is done by Alfred Hershey and Martha Chase. Alfred Hershey actually and Martha Chase used radioactive labeling trace bacteriophage. So a bacteriophage is a virus that actually infects the bacteria. So it used the bacteriophage DNA and also it used a protein. So it actually they labeled the DNA with phosphorus 32 and label the protein with sulfur 35. Why do they label them with these compounds, these elements? These elements are radioactive. They can easily glow. They can cause kind of radiation or kind of lighting, even when they are inside an organism. So the label bacteriophage was mixed with the E. coli, Escherichia coli, in the same bottle. So in the same flat bottom flask. So then the E. coli was also put with the bacteriophage that was labeled with a sulfur 35. So when these two were done, so they were put in the same flasks, and the E. coli and the virus were placed together in the same liquid culture medium. So in the flask, there is a liquid culture medium, which is going to make them to grow and have something to feed on. So what happened is the virus infected the bacteria and it injected the genetic material into the bacteria. So after that is concluded, so the mixture was agitated so as to dislodge the virus from the bacteria. So when the virus got dislodged from the bacteria, what happened is the content was being collected. So after the centrifugation, what was noticed is most of the 32 phosphorus was in the bacterial cell. That is to say, it got inside the bacterial genetic material, while the sulfur 35 was mostly found in the viral body or the container of the virus, but not in the bacterial cell. So what does that mean? The meaning is the genetic material, the DNA, went into the bacteria, but the sulfur did not actually go into the bacteria. So what was the summary from the test is the virus labeled 32P. So in fact, the infected bacteria, what happened is the labeled viral DNA was found in the bacteria. So the viral replication actually happened too. So there was replication of new viruses in the bacterial DNA. So new viruses contain this. So new viruses add this phosphorus 32 inside them. So that means they already incorporated it into them. So the liquid with the virus, there was no labeled DNA, there was no viral replication. But as for the group 2, in the infected bacteria, there was no labeled viral protein, there was viral replication, there was no virus. So new viruses did not have a label 2. But in the liquid, the labeled protein was found and there was no replication. So this would take us to the work of Erwin Chargaff, where he said he analyzed all of the A's and the G's and T's and C's in an organism, the DNA of an organism. So Chargaff rule says C is equals to G. That is to say, the percentage, the mole percent of C is very similar to the mole percent of G, and the mole percent of T is very close to the mole percent of A. The C, the G, the T, the A, the all nucleotide bases. The nucleotide bases which makes up the DNA. Now, Rosalind Franklin and Maurice Wilkins, they also did a very great job using the X-ray diffraction technique. So the X-ray diffraction technique indicated that DNA was double helix and also was twisted ladder. So the double helix shape was formed by two strands of nucleotide twisted around each other. So what does that mean? The meaning is you've got two strands of nucleotide twisted around each other that forms the one like something like this. This forming something like this. So another one twisted like this. So this is how the DNA was being formed. So James Watson and Francis Crick actually used the data by Rosalind Franklin and Maurice Wilkin. So they used the Franklin's and Chagall's data. So they use that to measure the width of the helix and the space of the bases. So then they use this knowledge to build a model that conformed with Franklin's and Chagall's data. So among the important features of the model constructed by Watson and Crick is the model has two outside strands, which consist of alternating deoxyribose and phosphate. So the meaning is you're going to have deoxyribose, then you're going to have phosphate, deoxyribose, phosphate, deoxyribose, phosphate. So the deoxyribose is a five-sided sugar. So you're going to have then connecting with the phosphate, then connecting with another ribose sugar with another phosphate. So this is what makes up the backbone. So while the inside is made up of the nucleotide bases, the cytosine, guanine, thymine, and adenine. So they're being paired like that: cytosine with guanine, thymine with adenine. So the nucleotide is they are subunits of nucleic acid. So the nucleotide consists of 5-carbon sugar, phosphate group, and nitrogenous bases. So the carbon sugar, the 5-carbon sugar, like this, ribose. So the phosphate group, OPO3, and the nitrogenous base, which is CTGA. So this is what makes up the nucleotide. So as we were saying, so the nucleotide is actually made up of these purine, adenine, purines, and pyrimidine. Purines are bases which are actually made up of double structure. So while the pyrimidine is made up of single structure. So this is the phosphate, this is the sugar, this is the base. So that's the reason why the C with the G and the T with the A is very sensible, because one purine would combine with one pyrimidine, one purine with one pyrimidine. The thymine would combine with the what? With adenine. Thymine is 1, adenine is 2. While the cytosine will combine with the guanine. And that would make the structure be very uniform. But let's say, for example, we have 2 purines and 2 pyrimidine. This would cause a larger space, and this would cause a smaller space. So that would not be even. So now let's look at the structure of the DNA. DNA often is compared to a twisted ladder. The rays of the ladder are represented by the alternating deoxyribose and phosphate. The pairs of the bases, cytosine, guanine, or thymine, adenine, they form the steps. Purine base always binds to a pyrimidine, like I mentioned to you before. So this is exactly what I'm saying. This is ribose, this is phosphate, this is ribose, this is phosphate, this is ribose, this is phosphate. So they alternate that way. So this is ribose, this is phosphate, this is phosphate, this is ribose, phosphate. So this is exotic. So this was adenine, thymine. This is the hydrogen bond between them. The hydrogen bond between them is this. So the hydrogen bond between the cytosine and guanine is 3, but that of the adenine thymine is 2. Why? Because guanine is actually having a double bond, got double compound. So there will be a connection between what there is going to be 3 hydrogen bonds instead of 2. So, and this is all we have to understand. So what is the basic building block of DNA and RNA? So if this kind of question has been asked in the exam, what should be your answer? What is the basic building block of DNA? What is the main thing that makes up the DNA? Is it all of this? It's nucleotide and phosphorus is nucleotide, because all of these are nucleotides. They've been made up of the cytosine, the guanine, the thymine, and adenine, all of this. So the only difference is RNA, the DNA has, just give me 1 minute. The DNA has C cytosine, as adenine, guanine, and thymine. But the thymine is not here. Instead, you have uracil. But the rest, we have it. So the nucleotide is the main thing that builds the DNA and the RNA. So if a section of the DNA has 27% thymine, so since we say A is equals to T, then 27% of thymine will be equals to 27% of adenine here. So how much is going to be cytosine? If you sum these two together, it's going to give you 54. So the remaining out of 100 is going to be 46. Out of this 46, half is going to be for cytosine and half is going to be for guanine. So if you divide this into two, it gives you 27%. So this is why your answer is most likely going to be B. So the next one, which was the conclusion of Griffith's work with Streptococcus pneumoniae? So it says the bacteria exposed to DNA can incorporate the DNA and change the phenotype. So which label represents the nitrogen bases? The nitrogen base is going to be C. What technique did Archie and Chase use in the experiment that led to the conclusion that DNA, not protein, was a genetic material in the virus? So what did they use? They actually used the radioactive labeling as an experimental technique to do that. So I wish you all the best, and I hope you would have a better understanding. Have a nice evening.